Have you ever wondered why you look like your mom or dad or why you're tall or short? Well, it's all about your genes. It contains deoxyribonucleic acid, or commonly referred to as DNA. It is the hereditary material that is found in our nucleus. It contains our individual cellular signature and determines eye color, skin color, and gender, as well as whether or not we have genetic diseases. More than 99% of our genetic information is exactly the same as any other information, but it is the 1% in which we all differ where things will get interesting.
We're going to give you a quick introduction into genetic mutations. But first, let's go over the central dogma of molecular biology, which is just the idea that genetic information in a cell is formed in the form of DNA. This DNA is used to generate complementary RNA through a process called transcription. The RNA is used then to synthesize a corresponding protein through the process of transcription. Looking at a quick example, our short DNA strand here will be used to generate an RNA strand. Remember that A pairs with U or T and C pairs with G. Next, our RNA will be used to generate protein through translation. Remember that during this process, RNA nucleotides are read in groups of 3, called codons, in order to generate corresponding amino acids. Generally, we say that mutations have the effect of making this synthesized protein not to turn out quite right. I'm going to give a quick shout out to sickle cell disease, which is an example of a disease that's caused by a genetic mutation.
You may remember that there is a protein in red blood cells called hemoglobin, which we can also call Hb. Hemoglobin is a protein that coordinates to iron ions in order to hold on to oxygen molecules and transport them throughout the body. The mutation that causes sickle cell disease results in a mutated form of hemoglobin called HbS, where the S is for the word sickle. The difference between normal hemoglobin and HbS is that one glutamate amino acid residue being replaced with a valine amino acid residue. This small change results in all of this mutated HbS protein aggregating together in a red blood cell, which makes it very difficult for that red blood cell to transport oxygen effectively. Just a side point, remember that red blood cells are initially generated from hematopoietic stem cells through the process called hematopoiesis.
Where are mutations found and how did they come up in the first place? So let's look at a couple of different possible mistakes that could lead to an incorrectly produced protein. First, we'll see what happens if a cell makes a mistake during translation. We'll stick with our example of sickle cell disease from before. Let's say that we have this sample piece of DNA with 3 nucleotides from the gene coding for hemoglobin. This DNA is transcribed to form the complementary RNA sequence GAG. That GAG would normally correspond to a glutamate residue during translation, but a mistake during translation might lead to a valine residue being translated instead to produce the mutated hemoglobin associated with the sickle cell disease. Notice that if a mutation happens during translation, the cell will only produce one mutated hemoglobin or HbS for each overall mistake. Since cells are making tons and tons of hemoglobin, just one mutated protein might not have that big of an effect on the cell. So we can say that mistakes during translation probably don't cause mutations like the one associated with sickle cell disease. Next, we'll look at mistakes during transcription. Again, we have our CTC piece of DNA, which would normally make GAG on RNA. But maybe a mistake occurs which leads to the transcription to a GUG instead, which would then code for the valine associated with mutated hemoglobin. If this mistake occurred, the cell would only make a few mutated hemoglobins for each mistake, since an individual strand of messenger RNA will only be translated a couple of times before being degraded. So we can say that mistakes during transcription probably don't cause mutations like the one associated with sickle cell disease. Finally, we'll look at mistakes in the DNA strand. If our CDC in DNA is mistakenly turned into a CAC, then our corresponding RNA from transcription will be changed and ultimately a valine would be produced instead of a glutamic acid. Now, since a cell's DNA stores all of its genetic information, that mistake would lead to all future hemoglobins produced from that gene being mutated. So overall, we can say that mutations will usually result from mistakes in a cell's DNA and not from the RNA or the protein. Next, where do these types of mutations come from? There are two ways a person can get a genetic mutation. The first is that they inherit it from their parents. Remember that DNA is passed down from parents to offspring. So if we have a mutated father here, then there's a good chance that at least one of the kids will inherit that mutated gene the same way that the child might inherit any amount of that parent's DNA. The other possibility is that the mutation will come on spontaneously, which is where a person suddenly gets a mutation in their DNA without parents having the same mutation. Spontaneous mutations can come from many different sources, with just an example being from DNA replication errors, environmental factors like certain poisons. It's also possible that genetic mutations can come on entirely randomly.
What is a mutagen? Well, a mutagen is any chemical substance or physical event that can cause genetic mutations. Chemical substances like poisons could also be mutagens. UV light or different kinds of radiation could also be mutagenic, and we classify mutagens into different categories. So let's say we have a person over here. A mutagen could also be classified as endogenous if it comes from inside the person's body, and it's a mutagen that's already found in that organism. But an exogenous mutation is one that comes from outside the affected organism, and it's something that's from the external environment. So, what are examples of endogenous mutagens? Well, the most significant endogenous mutagens are what we call reactive oxygen species, or ROS. And ROS are naturally occurring metabolites in the human body that are produced by mitochondria during oxidative phosphorylation. So, if we have this guy here who's about to chow down on a big meal, you can expect that during the metabolism of the meal, his mitochondria will produce a bunch of ROS like O2 that minus, which we call superoxide, which is an oxygen molecule with one extra electron as well as some hydrogen peroxide, which is another ROS that the body can produce. Now, reactive oxygen species, as you may be able to tell by their name, contain oxygen, like both of these examples do. But they're also highly reactive with different cell components, including DNA, and by reacting with DNA, they can actually cause significant damage to a cell's genetic code. One example of this type of damage is a double-strand break, and ROS can actually break a DNA's double helix into smaller pieces. And you can see why this type of reaction could cause a mutation, since it quite significantly changes the structure of the cell's DNA. The next type of DNA damage that ROS can cause is base modification, and that's when the nucleic acid bases are changed or swapped around, and that can pretty readily cause point mutations or maybe even other kinds. Now, you may be wondering, why would a cell ever make something that could damage itself? Well, it turns out that ROS actually have a couple of beneficial effects on a cell, and cells actually have a couple of ways to make sure that they don't cause damage. But sometimes, ROS levels get really high and cells can't deal with them anymore, and we call this oxidative stress. And antioxidants are something that your doctor might have told you that are good for you, and it turns out that part of what antioxidants do is help you make sure that ROS don't damage your DNA. Now, let's look at a couple of examples of exogenous mutagens. There are many different types of exogenous mutagens, but we're really only going to talk about two. Now, intercalators are one example, and one of them is called ethidium bromide, which you may be familiar with if you've ever done a PCR experiment before. And what ethidium bromide will do is it will jump into a DNA double helix and stick itself between the two strands. And when these intercalators intercalate into DNA, they can deform the structure of the DNA and cause some serious problems. Base analogs like 5-bromouracil, which we also call 5-BU, pretend to be a certain base, but then act differently than that base normally would. So in the case of 5-BU, it's an analog of uracil and looks a lot like it, but once it's incorporated into DNA, it can shift between two different forms. In its keto form, it will pair best with guanine. Now, if you're familiar with organic chemistry, you might know that 5-BU can convert between its keto and enol form through something called a tautomerization reaction. And overall, you can see how this base analog might be able to induce mutations in a DNA strand. The last thing we're going to talk about is what a carcinogen is. Carcinogens can be mutagens, but not all of them are. But in general, you can say that carcinogen is something that can lead to cancer, which, if you remember, is when cells in an organism divide uncontrollably and can form big masses of cells called tumors. Now, some carcinogens will work by making mutations in DNA that lead to cancer, but sometimes they might carry out their effects simply by increasing the rate at which a bunch of cells divide, without actually affecting their DNA. And some examples of carcinogens are tobacco, which come from cigarettes, asbestos, which used to be used as home insulation, and even UV radiation. What are the different types of mutations? Well, the first type of mutations we're going to talk about are called point mutations. Now, here I've just written at a random sequence of DNA, which is just a repeating pattern of CTC, which would code for a repeating sequence of GIG in the RNA strand, and finally, a protein sequence of 3 glutamate amino acids. So, a point mutation is when one of our DNA bases is replaced with another. So in this example, a thymine base is being replaced with another adenine base, which leads to a change in one RNA nucleotide and ultimately change in one amino acid. Another type of mutation is called frame shift, which works a little differently. So first, let's say I'll write out the same DNA, RNA, and protein sequences from before. But now, instead of changing one base to another, I'm going to add one to the sequence. And here, I've thrown in this extra cytosine base that I've written in blue. Naturally, this change would lead to an additional guanine base being in the resulting messenger RNA sequence. But what's interesting is that this mutation will change the reading frame of the RNA. Remember that RNA is read in groups of 3 or codons when being translated to form protein. But now, since we've added an extra G here, all of the codons coming from the extra G will look a little different. Now, instead of having 3 GIG codons, we've swapped out 2 for GGA codons. This means that 2 of our amino acids in the final protein will be changed, and in this example, they'll be changed from glutamate to glycine. So you can see that frame shift mutations usually have more significant effects on the final protein than point mutations do. Now, it is important to recognize that both of these mutations are classified and named for how they affect the cell's DNA structure, and are not really named for how they affect the resulting protein.
Now, our next type of mutations are nonsense mutations and missense mutations. Let's say we have a DNA sequence that normally generates RNA and codes for a cysteine amino acid. A nonsense mutation is any genetic mutation that leads to the RNA sequence becoming a stop codon instead. Now, missense mutations are a little different, and they're any genetic mutation that changes an amino acid from one to another. So, in this example, our mutation is changing. current transcript segment other resulting amino acid from a cysteine to a tryptophan. Now, you can see that nonsense mutations probably affect the resulting protein a lot more than missense mutations do, since that new stop codon that we're creating could chop off a huge section of the protein instead of just changing one amino acid to another. So, now we can divide the missense mutation even further into a bunch of smaller categories. Silent mutations are when the mutation doesn't actually affect the protein at all. Since many different RNA codes can code for the same amino acid, it's possible that the mutation might not affect the protein at all. So, in this example, CCA, CCG, CCT, and CCC in the section of DNA will all end up of coding for glycine. So, if you change the third base, it wouldn't affect the final protein. Conservative mutations are where the new amino acid is of the same type as the original. So, here, I have a glutamate and an aspartate, which are both acidic amino acids. So, a mutation that swapped out an aspartate for a glutamate would be a conservative mutation. Finally, a non-conservative mutation is one with the new amino acid is of a different type from the original. So, here we have a serine amino acid, which is a smaller polar amino acid, being replaced with phenylalanine, which is a large non-polar aromatic amino acid, and this would be an example of a non-conservative mutation, since serine and phenylalanine are different types of amino acids. Now, I'll point out again that all of these mutations are classified and named for how they affect the resulting proteins and aren't really named for how they affect the cell's DNA. So, let's look at a quick example. Sickle cell disease is a disorder where hemoglobin, or HB, which is a protein found in human blood, is mutated into a less active form, which we're going to call HBS, and it results from a single glutamate residue being converted into a valine residue. Now, we can classify this mutation as a point mutation since only one DNA base is affected, but we can also say that it's a non-conservative missense mutation since glutamate is being swapped out for valine. And the two are different types of amino acid since glutamate is an acidic amino acid and valine is a non-polar one. So, what did we learn? Well, first, we learned that mutations originate at the DNA level, but show their effects on their protein level, and second, we learned that we can classify different types of mutations by either their effects on DNA or their effects on protein. In reference to DNA, we have point and frame shift mutations, and in reference to protein, we have missense and nonsense mutations.
Today, we're going to talk about the overall effects of a genetic mutation and how mutations impact the organism as a whole. So, are mutations good or are they bad? And what kind of effect do they have on the affected organism? Well, there really isn't a good answer to this question at all. There are many different types of mutations out there that can result in big structural changes, like the little pictures I've drawn out here, or may result in little subtle changes that might go completely unnoticed. It's very difficult to call a mutation good or bad though, since it really depends on a huge number of things including the environment that the organism lives in. Let's look at an example of a good mutation. The bacteria Streptococcus pneumoniae is a bacteria that you typically see associated with pneumonia. One of the more popular treatments of pneumonia is giving the infected person an antibiotic like penicillin, which would help kill all of the bacteria and get rid of the disease. But sometimes, you can find some mutated Streptococcus bacteria that will have a special trait that makes them resistant to penicillin. And now, penicillin won't kill them as easily. It'll kill the bacteria without the mutation. We call this a good mutation because the bacteria are living in a human host where they are likely to encounter this deadly penicillin. Being resistant to antibiotics like penicillin would then be beneficial to the bacteria. Just to clarify, I'm calling this a good mutation for the bacteria, not really for the human infected, since it will be harder for them to get rid of the bacteria that are resistant to certain antibiotics. Let's look at an example of a bad mutation. The disease cystic fibrosis is usually caused by a mutation in the CFTR gene. I'm not really going to go into detail about how this mutation actually hurts you, but I'll leave you with the idea that what it does is it makes a mucus that you would find in a person's lungs really thick, which makes it really hard for people affected with the disease to breathe. In general, we can say that a mutation causing cystic fibrosis would be a bad mutation. But mutations aren't strictly good or strictly bad. In fact, there are some mutations that can cause some favorable and some disadvantageous effects. Sickle cell disease results from a mutation in a protein called hemoglobin that you'd find in red blood cells. This mutation turns hemoglobin into a much less functional form, which we'll call HbS. It's much less efficient at moving oxygen around the human body. Another effect of sickle cell disease is it makes the diseased person less susceptible to malaria. Malaria is a parasite that grows and multiples in red blood cells and can have a lot of nasty effects on the host organism. The malaria parasite can't really grow as well in red blood cells that are affected with sickle cell disease. In this case, the mutation associated with the disease has one bad effect, which is that the HbS isn't as good at carrying oxygen around the body, but also a good effect in that it makes it less likely that the diseased person will be affected by malaria, since they can't grow as well in the human's red blood cells. What did we learn? First, we learned that the effects of a mutation will usually but not always appear at the protein level. There are some exceptions to this rule. Second, we learned that genetic mutations can have advantageous, deleterious, or neutral effects depending on the type of mutation, the environment that the infected organism lives in, as well as a multitude of other factors.